Friends, welcome to Biology Made Easy. Today in this video, we will discuss the ultrastructure or the fine structure of chromosome. Ultrastructure, or we can tell that is the fine structure of chromosome. So earlier already we have discussed what is the chemical composition of chromosome. Chromosome usually consists of equal amount of DNA and histones. Chemically, it consists of DNA and histone proteins. Histones, they are present almost in the ratio of 1:1. And in addition to that, some amount of RNA and non-histones are also present. In addition to this, now the question is, why in the chromosome, in addition to DNA, the histone proteins are present? Because already we know that from a Griffith experiment followed by Avery, MacLeod, McCarthy, and that of the Hershey and Chase experiment, that it is not the protein, it is only the DNA that is genetic material. So if DNA is the genetic material, what is the necessity of these proteins to be present in the chromosome? So definitely they must have some function. So what is that function? Usually the proteins play a structural role in building the DNA into the chromosomes. That is why along with the DNA, the proteins are also present, which gives a scaffold or the structural component of the chromosomes. Now, earlier the view was that a chromosome consists of several chromatids. We know that when the chromosome in the dividing stage, prophase, metaphase, each chromosome has 2 chromatids. But earlier scientists, they had the view that a chromosome consists of several chromatids, and that was known as multi-nim theory. Multi-nim theory. That means the chromosome consists of several chromatids. But ultimately, with the use of viscoelastometry, it was found that the chromosome consists of only a single DNA, and this is known as uninim theory. Uninim theory. That means each chromosome consists of only one DNA. So according to the multi-nim theory, each chromosome consists of several DNA molecules, several chromatids, several DNA molecules. While according to uninim theory, each chromosome consists of usually a single molecule of DNA. And again, it was observed through this viscoelastometry method. And again, it was proved by also radioactive method. It was proved by radioactive method that it consists of only a single DNA molecule. Now, suppose we will take the cell of a human being. In case of human being, if we take the smallest chromosome, will take. And what is the length of the DNA in the smallest chromosome? The length of DNA is 1.7 centimeter. And if you take the longest chromosome of human being, then the length of the DNA will be 8.5 centimeter. This is in case of human being. And if the entire DNA of the human being will be arranged end to end, what will happen? You know that the distance between the two adjacent nucleotides is 0.34 into 10 to the power minus 9 meter. 9 meter. This is the distance between two adjacent nucleotides. And how many nucleotides are there in human being? In human being, 6.6 into 10 to the power 9 base pairs are there. So this is the number of base pairs and this is the distance between two base pairs. So if it will be multiplied, it will come to 2.2 meters. Now the question is, the size of the nucleus in case of human cell, it is varies between 5 to 8 millimicron. This is the size of the nucleus in case of human being. So how within the size of a 5 to 8 micrometer nucleus, how this 2.2 meters of DNA of the human being will be stored? If we will compare it, how it looks like, we can give an example. If there is a very fine thread of 40 kilometers, and this 40 kilometer fine thread will be folded to give rise to the size of a tennis ball. It will give rise to the size of a tennis ball. So this is as 40 kilometer fine thread will be coiled to give rise to a tennis ball. In the same manner, as 2.2 meters of DNA are present within this 5 to 8 micrometer nucleus. That means the DNA is folded around 10,000 times. Folded 10,000 times when it gives rise to the appearance of a chromosome. 10,000 times it is present. So, how it is possible that such a long DNA? Again, another example I will give. The 2.2 meters that is DNA which is present. If the DNA of all the 46 chromosomes will be uncoiled and they will be joined end to end, then the length of the DNA will be such that DNA of a single cell will move from Earth to Sun and again return back 100 times. That will say 100 times it will move from Earth to Sun and again return back. That is the length of the DNA. And if we will encircle it around the equator of the Earth, then it will be billion times around the equator of the Earth. So now the question is, how such long DNA is present within a very small nucleus? That we have to answer. So what will be answer? Usually the breadth of the DNA or the diameter, it is only 2 nm. The diameter of the chromatin. And how this is present? Now if we will break down the interface chromosome, if the interface chromosome is uncoiled, then it is found the diameter becomes that is 30 nm. And if it is further unfolded, then it gives rise to the size of 10 nm. So this DNA diameter is only 2 nm, but how this 10 nm and 30 nm structures are coming out? That means this is this structure of the DNA, which is the ultrastructure or the fine structure, and this is known as nucleosome. This is known as nucleosome. So the nucleosome is the ultimate structure of the DNA. And during the interface, how it looks like? It looks like beads on a string. Beads on a string. That means if this is a string and there are different beads on it, That means it looks like a beads on a string, or it looks like a necklace. Now here, this bead is nothing, it is a DNA. And these beads, these are the proteins. These are the proteins. So these beads are proteins which is wrapped by the DNA, and this is the chain-like structure, that is the DNA. That means both the DNA and the proteins, they combine together to give rise to these structures, nucleosomes, which are around 10 nanometer, and then give rise to 30 nanometer, then they give rise to fibrils and chromosomes. So what is the structure of this nucleosome? Let us discuss. The structure of the nucleus was first studied by Kornberg in 1974, and the name was given by outdated et al. Or co-workers, we can tell. So the nucleosome, the name was given by outdated et al. Means co-workers. When more than two scientists work together, then et al. What is now given, and it was discovered by Kornberg, 1974. So what is the structure of this nucleosome? Now already we have discussed that in the chromosome there are different types of histone proteins. What is the necessity of these histone proteins? Now as we know that when the DNA is formed and the different nucleotides are joined together, usually the phosphate or phosphoester bonds are formed. Phosphoester bonds are formed when nucleotides are joined together. These phosphoester bonds are negative. Now, if this DNA with these phosphoester bonds will be coiled to give rise to structures like the 10 nm size or 30 nm size, then these negative charges will repel each other. So how they will give a structure like this? That means it is not only the DNA which is coiled. So what happens? Along with this DNA, the histone proteins. These are the basic proteins. And the basic proteins, they have a positive charge. So the positive charge of these histone proteins that neutralizes the negative charge, and both the histone proteins and the DNA, they combine together to give rise to structures of 10 nm or 30 nm diameter size structures, which later on are called nucleosome solenoids. So the histone proteins, basic proteins having positive charge, these histone proteins, these are usually abundant with the basic amino acids like lysine and arginine. And depending upon the ratio between lysine and arginine, depending upon the ratio, the histone proteins are of 5 types: H1, H2A, H2B, H3 and H4. These are the 5 types. This is rich in lysine, while this is more rich in arginine. In all cases, both lysine and arginine there, but in H1 there is more lysine, while in H4 there is more arginine. And these two are much more conserved than the other ones. So how these histone proteins combine with the DNA to give rise to the nucleosome structure? Usually, out of these 5 ones, except this H1, the other 4, H2A, H2B, H3 and H4, they combine together. That means there will be 8 proteins. There will be 8 proteins. And what are the 8 proteins? That means 2 into H2A, H2B, H3 and H4. That means except H1, these 4 and 2 molecules from each one. That means this gives rise to the nucleosome core. It gives rise to the nucleosome core, or you can call that is the protein core, that is nucleosome core. Now this nucleosome core, it is wrapped by the DNA molecule. It is wrapped by the DNA molecule. And how many times it is wrapped? It is wrapped 1.75 times, or we can tell 1.3/4 times. It wraps. And while wrapping, how many DNA, how many base pairs are present? Now it contains 146 base pairs of DNA. It wraps. Then this DNA molecule moves, and again it wraps another nucleosome core. Again another nucleosome core, and again it wraps 1.7 times. Then again it moves, and in this side also again it wraps, and this side like this. And the distance between two nucleosome cores, this is known as linker DNA. Linker DNA, and it is usually 54 base pairs. 54 base pairs. That means this is one link. This is the linker DNA for this one. This is the linker DNA for this one. This will be for the next one, likewise. And when it surrounds, now it is tightened by H1 histone. That means this is the H1 histone. This is the H1 histone. And it is usually told that H1 histone is not a part of the nucleosome. That means now you see, here there is 146 base pairs of DNA, and in the linker there is 54 base pairs of DNA. That means in a nucleosome, you can tell that is 200 base pairs of DNA. So in a nucleosome there are 200 base pairs of DNA, 146 that wraps the protein core, where except H1, the other 4 are present from 2 proteins from each. So 8. This is known as octamer. This is known as octamer, or you can call it a nucleosome core. Nucleosome core. But nucleosome means from this linker along with this. So the diameter of this nucleosome, it is around 10 nm. So already we have discussed that when the interphase nucleus was unfolded, the structures having diameter of 10 nm was observed. So that is nothing but the nucleosome. And if further it is uncoiled, there is no other structure because proteins will be separated. Now these nucleosomes, again they will coil, having 6 nucleosomes in each coil. So again it will be coiled like this, and in this coiling there will be 6 nucleosomes. And now the diameter becomes 30 nm. This is 10 nm, this is 30 nm. So while this is known as nucleosome, this is known as solenoid. Solenoid. That means when the solenoid will be unfolded, it will give rise to the nucleosome. Now this solenoid is further folded to give rise to structures which are known as fibrils. That means now these will be further, the solenoids, they will be further coiled like this. They will be further coiled like this. Now when they are further coiled, here there will be addition of some more proteins. So during this, this is known as fibril, and this is 300 nm. Now that this will be 300 nm. That means 10 times it is increasing. And during this synthesis of this fibril, usually there is the addition of some scaffolding, structural, regulatory, and that is non-histone proteins. That means when the solenoids are converted to fibrils, here there is addition of scaffolding proteins, structural proteins, regulatory proteins and non-histone proteins, so that ultimately will give rise to the chromosomes. So now these fibrils are of 300 nm. Now these fibrils will be further coiled to give rise to chromatid, which is of 700 nm. So again this fibril will be coiled, and this will give rise to chromatid. It will give rise to chromatid, and it is about 700 nm. So the width of the chromatid is 700 nm, so that each chromosome has 2 chromatids. So the diameter of the chromosome will be around 1400 nm. Now if we will return back, that means each chromosome has 1400 nanometer diameter, and it consists of 2 chromatids, each having 700 nm. Now if this chromatid is unfolded, it will give rise to fibrils of 300 nm. If this will be further unfolded, we will give rise to solenoids having diameter of 30 nm. If the solenoids will be further uncoiled, then it will give rise to nucleosomes of 10 nm. If the nucleosomes are further unfolded, now the proteins and DNA will be completely separate. That means due to this folding of the DNA along with this histone proteins, and in the final stage some other scaffolding, structural, non-histone proteins are added, the long DNA molecules have been folded to be present in the form of a chromosome inside the nucleus. So this is known as the ultrastructure or the fine structure of the chromosome, the nucleosome model, the solenoid model, and what is the diameter of each one, and in the nucleosome how 200 base pairs are present, and which proteins are present in the nucleosome core and which one titans. All these things are very much clear, and the types of histone proteins. So now I hope you have a very clear idea regarding the ultrastructure of the chromosome. Thank you very much for watching the video. God bless you. Wish you a very bright future.